The overall goal of this procedure is to study the protective effects of melatonin using immunopurified primary human villous cytotrophoblast cells cultured under hypoxia or reoxygenation.
This method can help to understand the mechanisms and signaling pathways associated with pregnancy complication with placenta under hypoxia and reoxygenation. The main advantage of this technique is it is suitable to study the promising treatments such as melatonin on pregnancy disorder associated with increased oxidative stress.
Generally, individuals new to this method will struggle with villous cytotrophoblast isolation steps, such as mincing tissues and preparation of density centrifugation media gradients.
Though this method can provide insight into third trimester human placenta trophoblast, it can also be applied to other systems, such as human first trimester placenta and placenta from other species.
Visual demonstration of this method is important as many steps are difficult to learn without saying them. It can increase the reproducibility and reduces the learning time.
Demonstrating the procedure will be graduate students Andrée-Anne Hudon-Thibeault and Philippe Wong and Eugênia Assunção, a postdoc from my lab.
To begin this procedure, cut the umbilical cord alongside its placental insertion and keep them in 300 milliliters of tissue fixative solution for histological analysis. Then,